Hello everyone. So we are starting again our coffee break with TRANSFAC. And today we have a very good and important topic. It's about TRANSFAC download. So you can actually take the whole TRANSFAC in your hands and do whatever you like. And we have two guests today, and Dr. Volker Mattis, and he is a biologist, and he is actually expert in TRANSFAC. And we again having here Kamila Altenbergova, who is our IT specialist and developer of all tools to, different tools to work with TRANSFAC. And she was already here before telling about API, how to use API to connect to our toolbox. And today she will explain to you how to use different tools which we provide together with TRANSFAC in the package. And so then you can practically do many things like site analysis and other things which you listen today and learn. So please, your floor. Volker, Camilla.
Good morning, good afternoon. Thank you, Alexander, for the introduction. So I will start with giving an introduction into what kind of data are in TRANSFAC and how it's structured, particularly in the download release. And then in the second half, Camilla will take over and show how you can use this for making analysis for searching for transcription factor binding sites specifically. Okay, the TRANSFAC download contains eukaryotic transcription factors. They experimentally determine genomic binding sites and derived binding motifs, as well as data on combinatorial gene regulation and factor-factor interaction. Promoters, enhancers, and silencers annotated with transcription factor ChIP-seq, DNase hypersensitivity, and histone methylated intervals from the ENCODE project and other sources complement the manually curated binding site data. And then based on the positional weight matrices, transcription factor binding sites can be predicted in regulatory regions with the included tools or in the download by your own tools as well. And the download is specifically for bioinformaticians. It comes without an installation. You can just download and unzip the archives. Data files in DAT and JSON format. Promoters are also in GTF format. You have direct data access without a user interface. Data extraction is via Perl scripts or other programs which you write. Match and other tools are included for transcription factor binding site search. So the current versions, the versions which Camilla is telling about, they are Java-based. We also have an older C++ version, but that's more for people who already integrated that into their system. For use with, you can use with customer tools or incorporate them into your pipelines. Okay, the pictures which I show, they are of course not included in the download. These are pure text files. Okay, at the center of TRANSFAC, we have interactions between transcription factors and their DNA binding sites. And so these data are in the factor table or factor flat file, and the binding sites in the site table or site flat file. We also have interactions not only between transcription factors and their binding sites, but also between transcription factors, so between factors and cofactors and complexes of factors. The binding sites, they may be with or without a connection to a gene. So the genomic binding sites, they usually have a connection to a gene. And so via that, you can get a connection between a transcription factor and a regulated gene. But what's important is that we capture only those interactions between transcription factors and regulated genes for which we can can include a binding site. So position where relative to a gene where the factor interacts with this site and wire that with a gene, or the sequence, or ideally both sequence and the coordinates. So in other interactions between factors and regulated genes are not included. For that, you need to go to a pathway database, for example, our TransPath database, which includes all the derived factor gene interactions from TRANSFAC, as well as additional factor and all kinds of protein interactions. We have, in addition to the into the connection between a factor and a regulated gene, we also have a connection. So for monomeric factors, we also have a connection to the encoding gene, which is, of course, not necessarily the same gene as the regulated ones, but this is just because I show here only the flat files, not the individual entries. But however, there are, of course, also cases where a factor regulates the expression of its own gene, so that you can get also autoregulatory loops. And from these these interactions, so in a gene encoding a factor regulating a gene, and in case this regulated gene encodes against a factor, you can get the next encoded factor and extract a gene regulatory network. So in addition to these basic entries, so factor interacting binding site and regulated gene, we also have metadata, so the hierarchy of factor classification, which is based on the TF class by Wing and et al. And on the other hand, on the collection of binding sites for a particular factor, from this we can build a positional weight matrix, which can be used by the tools for binding site prediction. In addition to individual binding sites, we also include data from an originally separate database, the TransCompel database, so which captures composite elements, so the binding sites with synergistic and antagonistic effects. On the side of the gene, originally the binding sites are the coordinates of the binding sites are relative to a gene, so usually to this transcription start site, but can be also to other positions. However, for a number of organisms, for human and a number of other organisms, we include also promoter sequences with genomic coordinates, and to these promoter sequences, we annotate high throughput data, ChIP-seq data, as I mentioned, for the from ENCODE, for example, but also from other sources. And we also map the individual binding sites to these promoter sequences. And in addition to the promoter sequences, which are always for these promoter sequences, we always take a rough range from -10,000 to +1,000 relative to the transcription start site. But in addition to that, we also include enhancer and silencer sequences, particularly for human and also for some for mouse, and for example from Phantom. Yeah. Okay. I want to give a brief comparison between the download and the online version. So here for the online, I've taken there are various packages. The online version is available in various packages, and this here I'm showing specifically the omics version. However, in most regards what I'm mentioning here, the various packages are the same, but there are also some differences. Okay, first of all, the data, the core data, this transfer core data, transcription factor binding site, positional weight matrix, and so on, they are of course the same between the download and the online version. But yeah, the way the data are represented is different. In the online version, we also include various graphics and such kind of stuff, whereby the download is pure text files. And there are in the online versions, there are some additional data, which additional data are included depends on the package of the online version. But even in the basic version, the TRANSFAC basic online version, there are some additional data included which are not derived originally from TRANSFAC and the accessory databases TRANSPO and TRANSCompel, which you also get in the download. Okay, data access. Yeah, in the download, you have, after you download the data, or you download the data and then you have direct access to the data. In the online version, you get the access via an interface, via web interface or API, what Alexander already mentioned. And there are, we had already a couple of webinars, particularly by Camelia, on the APIs, if you're interested. And but it's the export option. You can export from the online version your analysis results and certain other data subsets, but you cannot download the complete data, all data. And particularly, and in the download, you have the command line access. And particularly what's important differentiation is the positional weight matrices in the matrix stat flat file. You can get them only on your computer from the download. In the online versions, it's not possible to download the matrices. So you can, in the online version, you can use the matrices only with the included tools. The tools you have in both basic transcription factor binding site prediction options for transcription factor binding site prediction, but in the online versions you have many more additional tools, and it's also so that when you're in the download version, which Camelia will show you, you need to submit the sequences in the data for analysis in form of DNA sequences, while in the online version you can also submit lists of genes and then So depending on the species, you can also submit a list of genes and then extract the promoter sequences for analysis automatically. And you can also do complete analysis of yeah, therefore it's called here omics. You can also do complete omics data analysis. And the analysis size, in both you can basically analyze whole genome, do whole genome analysis, and as I mentioned, you can do in the online version additional use the additional tools for further analysis.
Thanks,
Okay.
Volker.
Yeah.
Maybe before we
Yeah.
going further, you know, one thing I have to mention today, that of course you are free. I mean, I'm talking to our participants of this seminar. You are free to put your questions in Q&A. Please do so. I will watch what is there, and we will collect the questions. And if you have very perfect possibility now, Volker, as a specialist in TRANSFAC, can answer all your questions. And also, you know, we have often these questions, Volker, like maybe it's more about the first slide about the structure of TRANSFAC. So you have different. You mentioned you have different species in TRANSFAC, right? So
Yeah.
is it like every? First of all, what is this? Weights of those different species in TRANSFAC, and in TRANSFAC download? Do they all present, or they are split in different files? So how it is organized?
Yeah, so we have the different organisms. Particularly well covered is human and mouse. And in plants, the coverage is best for Arabidopsis, for example, and so on. But yeah, in principle, we are not limited to a certain organism. So it depends what kind of data. So the promoter data, they are restricted to a number of organisms, which I will show. However, the effector and the interaction with the binding site, there we are not limited to a certain organism, and we have several hundred organisms, but in some cases there are only few entries, and in case of human, the coverage is of course very high. Yeah, and this is in this factor dat and site dat, so it's for all kinds of organisms, or all kinds of eukaryotic organisms, I need to say.
All right, okay. But they are kind of marked clearly, so if someone needs to,
Yeah, and one important point is that everything is captured with the experimental details. So from which organism was the factor taken, from which organism was the DNA sequence taken, and that's an important point, that you also get data from cross-species experiments. So the factor and the interaction with a certain sequence or gene may not necessarily be from the same species. So, but you can get all this information and filter it respectively. So if you say, okay, I want not if I'm dealing with a human factor, I want only human sequences, human in binding sites that you can do.
All right, thanks, Walter. That's
okay. So here, that's just a screenshot from our web page. So just to show you an overview on the main TRANSFAC online packages. So in case you are interested in the online versions, you can find this information on our website. And as I said, I took here as main for the main comparison, the omics. Okay, this is now I have here a couple of slides where the download archives are shown and what kind of data files or the data files included in the archive. So we have here the main download archive, the main TRANSFAC download archive, and the data files, factor.site, gene.matrix, and so on. So in that format, and the most important ones are also included in JSON format. And here the fragment that contains the high throughput, the peaks or the fragments from the high throughput, the ChIP-seq data, particularly, and also some older ChIP experiment data. These are in a separate archive just because of the size, so it's easier to handle in a separate archive. And here this gives an overview or shows you the number of factor entries, whereby factor the factor flat file includes transcription factors as well as microRNAs. And again, so you have the entries, the factor entries in general. They have a type field, so you can see which data, which entries are transcription factors and which are microRNAs. And also on the binding sites, the binding sites for a transcription factor is on DNA, while at microRNA target site, that's typically on RNA. So you get both in the site but you get the type, there's a type field, so you can see if it's a DNA site or an mRNA site. So here are from this TRANSFAC main archive and also some interaction data, factor DNA site links, microRNA, mRNA site links, and so on. These are the two accessory databases which you get with the download. This is this transcompel database with these composite elements, so neighboring binding sites with synergistic or antagonistic effects. And here this transpro or promoter database. There are two archives here, one for the promoter sequence from human, mouse, rat, pig, rhesus, macaque, Drosophila, and Arabidopsis, and from rice, soybean, dog, chimpanzee, and plasmid. So I need to say this is actually, although you have these data also in the online version integrated with the TRANSFAC, the TRANSFAC core data, you get this, but we have two sources for promoter sequences. In the geneXplain platform tools, we use a different source. So there you have an overlap of what promoter sequences you can use. Either they are in the TRANSFAC interface or in the geneXplain platform interface. So there are some additional organisms which are not here included here in the download. Okay, here are the number of entries. So you can see the composite elements. This is a relatively small database. And here you see the promoter sequences, whereby this high number of promoter sequences, particularly for human, but also for mouse, comes from the fact that for one and the same gene, you may get a group of you have a group of transcription start sites from Ensemble, and based on that we do clusters, and for each cluster we include a promoter sequence, so there are more than one promoter sequences for a gene. Okay, so when you download, I show this as an example for the TRANSFAC main archive. After you downloaded the archive and you unpack the archive, you get this folder hierarchy. And in the DAT folder, you have these, as I showed already on this table, you have these flat files. And as an example here, how it looks like in one of the flat files, this example, the site flat file. So, okay, it's very small. Now you probably cannot read it, but you see there is the sequences is given here, and positions is given. Here is also the genomic coordinates given from the mapping to the promoter sequences, and here the source. So in the factor source in this case from the experimental data and the method which were used to show that this binding site was interacting with particular transcription factors. So you get there all the experimental data. And here it's in the JSON folder. You have here these flat files with the data in JSON format. And also here an example for the site entry. And just to show part of this description here, so we don't provide an installation with we do not provide an installation with the download. However, if you would want, you could also upload the data, for example, into a relational database, and there is a description for the JSON files, how you could upload them into PostgreSQL database, if you like.
So far we have, yeah, we got one question actually. So maybe we go for the questions first, and then we'll see what's time giving us. Hi, I have a question regarding the fragment table. Is it possible and how data can be filtered on the basis of experimental conditions of interest? For instance, the binding TF, cell type, organism, or treatment, in order to get all the list of binding sites found in these desired conditions. Well, the question is about the fragment table, but I guess this is rather about site table, but maybe both. What can you?
Yeah. Okay. So for site, yeah, you can in principle filter. There is a relatively large number of there are these methods. So you have seen okay. Or maybe I don't know if you see, yeah, basically here, so as I said here, this MM fields, that's for the method, or for example, jail retardation or so was used for by seeing the binding site. And you can see there may be for one and the same binding site or interaction between one and the same binding site and a particular factor, there may be various methods which have been used, and you could in principle use this for filtering the sites. And also the factor source. And here in the factor source, you see, for example, that these are cell lines from which an extract was used. So the protein extract was taken from the cell line, and in some cases it may have been induced by something. For example, in this case with interferon alpha. So the cells may have needed to be induced by interferon alpha to have the particular transcription factor in the extract, and then the extract was taken and binding was investigated, for example, with the gel retardation assay on this sequence.
And the
So
factors,
yeah.
And you can also filter by the factor, right? Transcription factor. So
yeah. You can, I mean, yeah, actually that's I can show in the slide which will come in a minute after the question. So it's possible, but yeah, it's you need to look what's the crucial thing or what you are interested in.
Right.
And for the fragment, it's in principle similar. You get these data, I mean, when you know the data from ENCODE, for example, so you see a certain data set where the peaks or the binding fragments where transcription factor is found bound to in the genome. This depends on on the transcription factor, but also on a certain state on certain tissue and certain condition. And there you can also do that. So for some factors, you may get in ENCODE, and so only only for one cell type and one condition of all the peaks, but for some factors you get it's for, I don't know, 50 different conditions.
So it depends on how this factor is well studied. Yeah.
Yeah.
Right.
And
all right. Okay. So maybe you can show those slides you wanted just to keep also in time that Camilla will tell us about the software, how to use all this data.
So I just wanted to show a little bit more in detail. There are I think 6 or 7 slides which show in a little bit more in detail how the entries are interlinked. So here is an example. This from the side entry again, and you have a link in the side entry to a factor. So in red here, the binding factor, and in blue here, the gene to which this binding site belongs to and which is regulated via that. And here, yet now you have here this corresponding factor entry in red, and in blue the corresponding gene entry, which are linked via this binding site. In green, the link, but we also provide a shortcut between the factor entry in factor.dat and the gene entry in gene.dat. So you can use this shortcut, but as I mentioned, it's important that the binding site, so this interaction is only there if we can provide a binding site. Here, this would be between two transcription factors, a factor and an interacting factor, and also to a factor complex. So a factor, a transcription factor, may interact with a cofactor. So where one factor is binding to the binding site, but the cofactor is only interacting with the transcription factor. However, often it's also the case that this factor complex maybe form a stable complex, so which you can show via co-immunoprecipitation, for example, in absence, which is stable in absence of the DNA binding site, and the whole complex interacts with the binding site. And so where you have often it's the case that not a monomeric factor, but a homodimer or heterodimer interacts with the binding site. And this is a different case. This is from this compel from this composite element database, where the two factors do not form a stable complex in absence of the DNA, but this interaction between two factors binding to their unique binding sites can and the two factors, the two neighboring factors, then interacting with each other, this may stabilize the whole complex and lead to synergistic or the opposite case to antagonistic effect of these binding sites, these composite elements, which if you have heard one of our other talks by Alexander also, then you know that we use these not only these data from this database, but this fact, this that these factors interact with these others. So composite modules, predicting composite modules, we use this often for our analysis. Okay, here, this is the example. In this case, not the site DAT, but fragment DAT. So from these high throughput data, as I said, we have these fragments from these ChIP-seq data, particularly in a separate table. This has several reasons. One, first of all, is because the data is a very big amount of data. So it's better to keep them separate from the site entries. And but there are also qualitative differences. One is that these fragments, they are relatively long. They can be several hundred nucleotides long, while the normal binding site entries are usually much shorter. There are a few exceptions, but usually they're much shorter. And also the association with the gene, we get these fragments, these ChIP-seq fragments, without a connection to a gene, and we map them ourselves to genes by distance. And in case of the binding sites, there is we get from the literature, usually based on functional interactions, also a connection to a gene. And here for the microRNAs, as I mentioned, the factor DAT table includes transcription factors and also microRNAs. So you can or need to filter by the type field. Again, that's if you want to separate them, what's usually the case. And here the site is usually on the mRNA and not on the DNA. So the site entries you can also filter between RNA sites and DNA sites.
Thanks a lot, Volker. All right, so you see how complex the life is. So let's then maybe switch to Camilla, who will tell us about how to make it simple, how to take all this information and use it in the software to solve your questions.
Yes, sure. So hello, everyone. I will take it from here and talk about one particular part of the TRANSFAC download package. It's the match library and the tools that come with the library. So let me start also with a brief introduction first. Okay, so the main the main tool of the TRANSFAC download package is the match tool. So what is the, as Volker just presented, so what is great about TRANSFAC? We have the database with the information about transcription factor binding sites. And let's say that you have some sequence and you want to search for these binding sites on your sequence. So this is where the match tool comes and is useful. So what it does, so we providing that you have some sequence on which you're looking for transcription factor binding sites for particular factors, having the download package on your hands, you have the access to the whole TRANSFAC with all the positional weight matrices. You can create the profile, or you can select the collection of the transcription factors you want to look at, and then using the match tool, which will compare the binding sites with the sequence that you have. It will predict the possible sites on your sequence. So this is like the most important task, probably like most common, why you would like to use the package. And what you get when you have the download package, you have one of the Volker already showed you the structure of this download archive, and one of the folders were match library here, and this is everything that you need to run the match locally on your computer. So again, as it was mentioned, at the moment we provide match written in Java. So you would need you'll need Java on your computer, and also probably because this is command line tools. Again, as we compared with the online version, we have online version of the TRANSFAC and of the tools available in our geneXplain platform, and there are also separate TRANSFAC tools which are also available online, and they come in a web interface, maybe a bit easier to use, to get familiar with. And this is more for our hardcore users who want the download package and want more control over how they use the data. And also okay using a bit more complicated tools as a command line, and maybe more familiar and comfortable using this more advanced maybe way of interacting with the software. So what we have here, we have several jar files. So this one is the main one, match library jar, so which is actually the executable with the with the tools. There are some dependencies also, and I will show how to set it up, or your environment to use all of this. And also there are some README as a documentation, archive as documentation to look at, to have some help with your work with these tools, and also some again additional parameters for the match that are needed to provide you with the very good quality results on your search. So there are just some parameters that are pre-calculated and saved for you. You can read more again about match. We have the paper published on it, and there is more information on our website about how the tool works and what is the logic and algorithm behind it actually. Okay, so yeah, as I said, this is the command line tool. So you'll need to interact it via command line. I'll do a bit of demo today just to show how it actually works on practice. I hope it will be not too difficult to follow. It's okay if you have any questions, please write to our chat. We'll try to figure them out. So yeah, I'll go in bit more detail about each command now. But just before going there, you'll see that here I printed the available applications within this match library, and you see that there are actually different different sub-programs, some small applications within the library to do some specific tasks in preparation for running the main match tool. So why there are several tools? Why would you might need to use some of them? So there are tools to to work with the PWM libraries, with position weight matrices libraries, because again, as we said, we have the TRANSFAC specific DAT format in which you have the sites, right? Or you can also convert to JSON, and then working with the JSON might be more also more convenient to some users, or you can also might want to convert it to some other formats to work with some other software. So just for this flexibility, it's possible to make a specific JSON files with the specific position weight matrices, or extract some models. And then also it's possible to do some profiles. And for us, the profile means some, again, like some specific collection of the matrices, which will be then used with match profile. So match needs to, when you tries to predict the binding sites for your sequence, some references needed, right? Like what to look for, with what to compare. And of course, TRANSFAC has, as we showed, a big collection of different matrices, and just in order to tailor it to your specific needs, to maybe your specific species, or with like to play with the cutoffs, and just select that collection you want to search match against. We create this profile file with some selection of the matrices. And so there is also, again, comes the tools to prepare such profiles before running match. So this is again just an overview. We'll see how all of this works. And just to mention very quickly, there are sometimes requests from our users that they might want to work with other formats rather than just TRANSFAC matrices or JSON. So there are actually several tools you can use. So that's the beauty of having the download package, that you have all this data on your hands. So you can write your specific, your own scripts. For instance, here we propose using Python library, writing just a small Python script with the maybe bioinformatics library available for Python. You can already work here with a lot of different formats. So you can read the TRANSFAC file format from the DAT files you have in your package, and then convert it to some other formats you might need with other software. So you might find actually some other more convenient ways or programming languages to work with, but just for you to know that there is like such a possibility, I wanted to mention this. So okay, this is the just the introduction to specifically match library. And now I would like to turn to demo, a bit demo about how this now actually all works in practice. And here I wrote some commands for me, just that I do not spend too much time now of your time while typing all of this. So I will go command by command, and we'll just quickly explain what I'm doing on each step. So for now, you see that I have locally on my computer, I'm currently this match library folder, subfolder of my download package. And here you see the same files I just mentioned. So I have some jar files, I have some additional database file here, which match will use, and just one more additional file. This is just a faster sequence with some short sequences for me to demonstrate. I will use this file as input for match. So this is the contents right now. And first of all, what we need to do to start working with the library, we would need to declare some variables in our environment. So specifically, that's this is two, you can name them whatever you want, but the match will need to know which where his dependencies are, where to look, and just some passes for dependencies and for matchlib itself, because on your computer, of course, you're gonna have it somewhere on your system. So for me, this is the path to the folder I'm currently working in, and here just this is for more convenience, because not to use every time the whole path to these jar files, and just having it as a reference here as a variable is more convenient. So now I will clear my screen a bit so that it's not too confusing. So first I wanted to run very simple command. So here, as you can see, I'm using Java. I have Java installed on my computer, and using these variables I just declared in my environment, I am actually running this match library jar file, and this is the main class here, match library app, from which everything starts. So you mainly interact with this class. Having this flag H means that we want to print the help for the application. And here already you can see the list which I already showed you on screenshot. So we see the available applications, and we see some descriptions about each of these programs. So this is a very good start, just to print some information and see where you want to go further. And as you can see, for more information to this match library app, we just add the application name from this list, again putting this H flag here, and you're gonna get some more information about specific application within this library. So let me clear again, and let me show you. Okay, so you see here now I edit match. I want to know more about how to run match, and I'm also having this H flag here, and you see the parameters. So what are the parameters to run this tool? So if you're familiar with how command line works, this probably should be very familiar to you. So you just have some flags, and then you specify here the paths to your files. And as you can see here, to run match, so this is our ultimate goal. We want to provide our sequence and then see what match can give us. And we can see that we will need some more files for that. We will need some profile path, so we need to have our profile. We need to have our FASTA file, and then this database with scope parameters I mentioned, we have it here already, but we need to also specify all of this in order to run match. So let's prepare everything we need. So I'll start with this. So here we have something TRANSFAC model files in JSON format. Okay, we have a specific. Okay, so now it will run. I will tell you what is going on. So we have this command here. So again, I'm just running match library app, and we have this make JSON tool. So what it does, we take that file again from our installation. So in my installation, I have that subfolder, right, with the matrix.dat file, which was already mentioned by Volker. And I'm just outputting the JSON file. I'm just converting that to JSON, because this is what is required by our match. So I run very quickly, and here, so if I clear again, and if you see, I have a new file here called matrix.json. So now we just have our matrix that converts it to the format we need. So what do we do next? Then we okay, so I will show another command. Okay, this is just for demonstration. So now using this matrix JSON, I can run some other tools. For instance, I can run extract models. And let's say I want the model for specifically just one transcriptional factor, P53. And then having it on hand, so having matrix JSON, I can just extract one specific model. Let's see what we have here in JSON. So here we'll have information, you see, just one model in JSON format. So of course here you can provide more models. You can provide a list also, actually, which will be like more convenient to extract some specifics models if you need it. If you need maybe to use it then with some other software. So this is the command just for the demonstration. Let me clear the screen again. So now again, I have the matrix JSON, and I can use this matrix JSON to create a profile. So now I want to create a collection for like a specific collection of matrices for which I will then do the match search. And this is here I run profile type list program tool. This just gives me what are some types of profiles I can create. And here you can see that depending on what type of profile I'm choosing, we have some different cutoffs for the prediction. So how the models are selected for further match. And this is just again like just some information to further create the profile. So in order to actually create the profile, again, let me clear the screen. Okay, so now I have another command which is called matrix profile. And here I select I select the type of the profile I'm creating. I'm selecting some cutoffs for the quality of the matrices which will go into my profile. I'm selecting that this is going to be a human profile, so I'm only interested in the human matrices. And here just some more information about what JSON. So here all my matrix that was used to create this JSON file. So we'll select some matrices from the whole selection of what we have in the database. And yeah, some helper things like this is the path to this database I mentioned before, and also just the name of our profile here. So as you can see, it here it will what this tool will do, it will go through the matrices we have here in our JSON, take the human matrices and check if they actually achieve our p-value cutoff here, which we are interested in for the match. And you see that if there are some some matrices will not be good enough for our profile, so we'll just skip them. And our profile will have just a smaller number, so it will be smaller number, smaller selection of the of the matrices. Okay, so we are progressing here. It doesn't take too much
So
time.
while it's running, maybe you can explain why it's actually sometimes not achieving a cutoff. Do you know?
So because we have this database with the pre-calculated scores for the for each matrix, right? So for matching, and we have also some pre-calculated scores and p-values for specific sequence. So it can be that here we want maybe only sequences with very good p-value, with very good
So simply these
s-
p-values cannot be achieved for these metrics, right? So
yes, so it was not achieved. It means that maybe if you do so depending on how very specific you want your search to be, you want to set very you saw there are different like settings. You can choose profile for scores, or you can choose profile for p-value, right? So depending on how you want your search to be strict, how good quality of a match you are searching for, you can regulate how you build your profile, so how restrictive it is, or maybe you can make the p-value maybe a bit higher, and then or maybe like scores maybe a bit low, and then you're gonna have your profile bigger, so you're going to search for more matrices during the search.
exactly, yeah,
Yeah,
right.
but
So,
so it depends on what is your goal. So if you are okay with the having much looking for binding sites of like more matrices, of more transcription factors, you can do that. Maybe you can later then in your further analysis somehow narrow your results, right?
exactly.
Or if you want already to have some more stricter, some very good quality matches, then your profile will be maybe a bit wilder. And so here now we have our profile generated. So we have here a new file called profile, and we are actually now ready to run the match tool.
All right, so
finally. Because
let it run, but I guess it will run. But we have a very short question here in between. So is the matrix JSON file created with a command line different from the one which is which you can download from the package, or it's the same?
I think actually it's the same.
Okay.
I just wanted to show that maybe you will you probably maybe you filtered your dat file, or you also do like some sub selections or something. So I just wanted to show that
Exactly.
because with the match you need your matrices in JSON format. So this is how our this command line match works. So just in order for you to know that there is possibility to convert in any dat file, or if you first have like your positional weight matrices in some other format and you want still to use the match, because it can also be constructed like maybe not necessarily use only TRANSFAC, although TRANSFAC is very good, but maybe if you want to add your own positional weight matrices and you then want to run it with the match, you need to convert to JSON. So just to demonstrate that you can convert to JSON, but basically, yes, so this we already like the whole matrix dat and matrix JSON, they are the same information in different formats. So you already have it prepared for you in the package, actually. Okay, so what's going on here? Again, this is the first very long part is just calling for our application and then running our match tool here. So first we say where to save our results. we provide path to our profile. Right now, everything for me is just in this folder, so I don't need to write long path. I just have the name of the file, and here again we have this pre-calculated parameters for the match here in the same under the same path. And I already mentioned this is my FASTA file with the sequence, which is actually the input. And the referenced initial matrix JSON we used to create our profile. Okay, so here we were successfully connected to the parameters. And now if you look at the contents, we have a new file here called match result. And if we open this match result file, you can see that we have here the information about the possible matches. So we did the here's some information about how this run was created. These results were created with what files, if you forget later and come back. Here you have this information, and here you see the matrices, and you see the okay, you see the okay, because I had in my FASTA file several sequences. So here you have the information about okay, so this is for the results of match for this sequence, for your reference to find it like from your FASTA. And here you can see the actual predictions in nucleotides here. So where the match of the specific binding site was found, and some statistics about the match. So this is in text format. You can then, of course, convert it to any. So this text file, you can do whatever you want with the format, uploading it somewhere, using it further, or just also to filter if you want to look at the results more closely. But this is just to show that yeah, this is
so
usually the
you see the column, you see the position, and what is this plus minus? You maybe you can explain a bit.
So this is the strength. So the position. So the chromosome and the strength. So minus plus strength.
All right. So in which strand of DNA you found the site? Yeah.
Yes.
Okay.
Yeah. So this is yeah, so again, depending on what your needs are, because usually our customers who have the download package, of course, they are again like more advanced users who really like the command line tools where they can then integrate them in their own scripts and their own pipelines and have more control about, because the simpler is the format, actually, the easier it is to customize and to add to your pipeline. This is just a little demonstration. I just run it all in command line. Of course, you can also integrate it, write some scripts where all these commands will be run for you. You can then reproduce it later. And because it's on your own environment, on your own computer, it's more controllable, because with the online tools, while of course having constant updates and new changes are great, but like in online version, we try to also have like some archive version of the databases, but sometimes online versions, they can be updated. Sometimes for reproducibility, there might be some problems that you might need to adjust something when you come back to your older results or something. And also one of the advantages of having everything on as a download package, because you have the whole control, you have it all on your computer, in your environment, and then you are sure that you can reproduce your results in whenever you need.
All right,
So
I see.
yeah, so this is all from my side. So I just wanted to demonstrate here how to work with the matching command line tool. If you have any questions, please let me know.
Yeah, we have one question again. So in TRANSFAC DB, do we have data cataloged separately in brackets for A, specific diseases causing genes and their TFs, for instance, diabetes, hypertension, different cancer types, B, for certain viral proteins interacting to human genes, for instance, AC2, SARS-CoV-2, of course, spike protein and the binding TFs to human ACE genes. So quite a long question. I'm not sure, maybe Volker can answer this better. Volker, can you?
Yeah, so we don't have disease data, not in TRANSFAC, but regarding the viral data, in principle, we include viral information. I mean, for example, using a human transcription factor in when a human transcription factor is used by a virus for expressing its gene, then we may include these as binding sites for the human transcription factor, so the binding sites on the viral genome.
And the other way around, perhaps.
Yeah, the other way. Principle, yes, but I think it's more rare. It's usually more so that the virus uses human or host transcription factors, or maybe even So in
well, some viruses can encode a transcription factor, right?
That's in principle also okay. That we take the viral transcription factor and the viral binding site.
All right, and then was a question in the human genes like ACE, like a receptor, and of course we have sites for different transcription factors in the promoter of this human gene. So although it's play a role in viral, biology, like an entry point for the virus, but expression of that gene is controlled by human transcription factors. Some case in case of COVID, or it's better, the name of the virus is this long one, SARS-2 virus is actually it's as far as I know, it doesn't encode transcription factors, but actually it encodes some proteins which activate signal transduction pathways in human cells. For instance, they inhibit, in fact, the interferon pathway, and this way it influencing the expression of the genes which are controlled, whose transcription factors are controlled by those pathways. And that's actually a level of complexity which may be not easily achieved when you have just download TRANSFAC. Well, maybe you can touch a little bit. So Camilla, you are so fond of download TRANSFAC, and I perfectly agree with you. Of course, when you have everything in hands, you can do whatever you like. It's the title of our talk today. But what could you say, briefly, what you are missing actually when you just have download, but you don't have a full access to online tools and or even integrated tools with other databases? Just briefly, can you say couple of words about that? So what you're missing when you have just download?
I mean, do you mean in comparison to what we provide in the geneXplain platform?
Exactly. So
And
although
also
you show,
which accessible via API also?
well, yes, like you mentioned that, for instance, the access to pathways, to access to diseases, there was a question, right? So whether we have in TRANSFAC download the disease information. No, we don't have, but if you, as I understand, if you have an integrated access through the online, you can get the disease information, right?
Yeah, so we in the online version, we have depending which package you're working with, and there are packages which include disease data. So disease data in this case is biomarker data. So which genes are connected with what, with which disease, and you can use this also in some of the analysis.
All right. Yes. And but coming back to the, all right.
But the directly, so the direct mutations of transcription factors or other proteins we do not directly include. So you can do a functional analysis. I mean, you can map genes to diseases. That's possible, and in parallel analyze them for for mapping to pathways and so on.
Yeah, right. So I think we will make a separate coffee break to talk a bit more about this direction. So once you have your transcription factors, so what is next? What's next?
Yeah.
So you have to connect it to diseases, you have to connect it to pathways, and what can you do for that? So we'll talk about that later. But for today, maybe just a final thing, coming back to the download and this command line, Camila. So how fast is the tool? I mean, you showed that you just run the whole library on one sequence, or how many sequences there were?
Yeah, I think there are like 3 or 4 sequences, not very long.
Yes, but
So
if you
that's
run
why,
it
of
over,
course,
over the whole
yeah.
genome, for instance, can you do that, or can you try it? How long would it run?
Of course, that will then depend on your computational abilities of your computer server that you have. This is also one thing that probably if you have our download package, not just on your personal computer, but maybe in your universities or your company's premises, on your servers with the dedicated computational power, then of course that will also like be maybe faster than if you run like a lot of analysis even in our online platform, right? On our online tools, because of course we have a lot of customers, and then the computational power is divided between our customers, right? So that's also one thing worth mentioning, that yes, if you have the capability, if you have a possibility to have a lot of computational power and then running long sequences, so doing match for long sequences with the like with the maybe even whole genome, and then of course it like it will be faster than online, right? But overall, of course, it depends on the mostly then on your software, because this is now your environment, so it will depend on your environment. But as you can see right now, so just but even if you just work on your PC as I did just right now, so for some specific sequences, so I had faster, I think 3-4 sequences, it's also doable. Might take longer time than, of course, than if you have a server dedicated. Dedicated server, but still it's possible to run even on so it's not very it's not very computationally heavy task. It's just that if you have more computational power, it will be faster.
Perfect. Okay, so I guess thanks a lot for your very good, introduction to the TRANSFAC in general, and also for these tools, how you can easily run the match to find your sites. We are online now a bit more, quite long today. So if you guys have more questions, just give your last question. You have a last chance before the end. One last question. Well, very short. Can match be run in parallel?
I think that can be like I'm not sure. In parallel, you mean in parallel on several sequences, like in several scripts? Then it will depend on how you how you run it, right? So it is possible, of course, like at the same moment to run match on several sequences, like on several FASTA files, maybe at the same time.
No, what I think they mean rather the if you have many cores on your in your server, does match actually using these parallel computational possibilities using several cores? I guess yes.
Yeah,
to a certain extent, right?
because this is Java, which
Yeah.
is running. So yeah, so Java I think can be run in parallel, so it will take advantage of your setup. So yeah, so again, our current match which comes with the package, it's just a Java program, so just
So you can
you're running
configure
Java
Java.
application.
You configure
Yes,
Java for
you
parallel
configure Java.
computation. Yeah, right. Okay. Okay, with that, I think we can finish for today. And thanks a lot, Volker and Camilla, for your very valuable comments and the introduction. And see you next time. Goodbye.
Thank you.
Goodbye.